तो कहाँ से आएगा फिर ये ये देखिए, और यहाँ पे ये एड कर दीजिएगा रेड, ठीक है। ऑन Good morning, all respected members, faculty members, and my dear friends. Today, I am going to present my journal club, and my topic is uh, Chlamydia Psittaci infected dendritic cell communicate with NK cell via exosome to activate the antimicrobial immunity. And my moderator is Dr. Bimal Kumar Das. And my moderator is Dr. Bimal Kumar Das. And first of all, introduction. And Chlamydia psittaci is an intracellular gram-negative coccoid bacterium that infects both birds and humans. In 1879, psittacosis or parrot fever was documented for the first time by writer, and in 1893, when transmission from parrots to human of an infectious agent causing flu-like symptoms was observed by Morange, and agent of psittacosis was finally identified in human by Berson et al. In 1930. During the winter of 1929 and 1930, a pandemic of human psittacosis occurred in the United States and Europe. And psittacosis can affect any age group and genders. Incidence tend to peak in middle age with an age range of 35 to 55, and the mortality rate is 15% to 20% in human psittacosis case, and the mortality has been extremely rare since the advent of antibiotic treatment. Recently, other bird and animal species have been gaining more attention as a potential source of human psittacosis, such as poultry species, pigeons, and even mammals. Due to absence of an electron transport system, lack of cytochromes, and the apparent inability to synthesize ATP and GTP in the same way as rickettsia. Now, because it's initially classified as a parasite, but on the basis of these things, it is classified as a bacteria, and it is the another feature of relevance to the bacteria is divided by binary fission. Cell walls possess DNA containing nucleoids without membrane between the nucleoid and the cytoplasm, ribosome with antibiotic susceptibility characteristics. And Chlamydia are considered energy parasite since they depend on the eukaryotic ATP for their replications. It depends on human ATP, yes, cell ATP. Predominantly, two alternating chlamydial forms are present, that is elementary body and the reticulate body. And infections of susceptible cells is mediated by the elementary bodies. The size is 0.2 to 0.3 micrometer in diameter, and the bacterial multiplication within the cell is carried out by the RB, a particle of 0.5 to 1.3 micrometer in diameter. And this is the chlamydial development cycle. Attachment and entry of the body, and these are the mechanisms which are involved for the attachment and entry of the elementary bodies. And the second is primary differentiation, MAB activation, chromosome condensation, and early gene expression for the development of division of cells and the inclusion modifications. And these are the next is the cell division. 6 to 14 hours have lag phase and peptidoglycan involvement, and then again these are the two pathways. First is abnormal growth, that is goes to for the persistence, and the another is the RB development of RB bodies, and is the rapid multiplication. Maximum transcription translation occurs in this phase. Inclusion expansions also occurs in this phase. Effector secretions, all these bodies, persistence and RB bodies again convert into reticulate inclusions bodies, and that is secondary differentiation. And in this case there are many gene rearrangement is occurs, and RB and EB release and reinfections again to the nearby cells. Transmission of psittacosis. Common reservoirs of Chlamydia psittaci include wild and feral birds such as seagulls, ducks, herons, egrets, pigeons, blackbirds, grackles, house sparrows, and cladder. And people become infected through inhalation of aerosolized bacteria when exposed to infected birds, or by the handling of contaminated feather, dander, fecal materials, or body tissue. Beak to mouth contact also occurs. In human to human transmission, while possible, is believed to be a rare event. Human psittacosis is mostly linked to parrot or ornamental birds as source of infections for human beings. And the disease, the Chlamydia psittaci causes a disease called psittacosis. It is a zoonotic disease and also known as parrot fever or ornithosis. And the incubation period of Chlamydia psittacis in human is 5 to 14 days. And common symptoms of psittacosis include abrupt onset of fever, chills, headache, malaise, non-productive cough, and dyspnea. Rare complications also include myocarditis, encephalitis, hepatitis, conjunctivitis, acute respiratory distress syndrome, and multiple organ failure. And this Chlamydia psittacis could possibly be linked to development of ocular lymphoma. A number of severe cases of human psittacosis, that is atypical pneumonia, were reported recently in human beings. The diagnosis of psittacosis, culture, bowel, sputum, pharyngeal aspirates or swab are considered as the most best materials for the culturing of diagnosis. And there are many tests for serological tests which is used in diagnosis of psittacosis, that is complement fixation test, enzyme linked immunosorbent test, microimmunofluorescence test, and whole cell immunofluorescence test, and the immunoperoxidase test. And there are also molecular tests for diagnosis of human. PCR and real-time PCR also can be used for diagnosis, and as well as the differentiation between the different species of chlamydia. And treatment and prevention of psittacosis. Chlamydia psittaci are sensitive to both microlides and tetracyclines. However, tetracyclines are the drug of choice. Microlides are the drug of choice for children. People with more severe presentation should be treated with doxycycline. And patient isolation, prophylaxis of contact are usually not indicated, as person-to-person transmission of psittacosis are rare. Timely diagnosis and reporting may aid in identifying the source of the infection and the controlling the spread of disease. Educate patients and pupils about the importance of handling the birds and cleaning of birds' cage safely. Now I am coming in the my topic. Chlamydia psittacis infected dendritic cell communicate with the NK cell via exosome to activate antimicrobial activity. First of all, I want to explain exosome, because exosome is the main key for the induction of NK cells in to activate the antibacterial activity of chlamydia cases. So, exosome released from dendritic cell are called exosome released from dendritic cell are called dexosomes. Membrane enclosed vesicles of endosomal origin that are secreted by the most cell type, and play an important role in cell-to-cell communication between the cell-to-cell communication in blood cells and epithelial cells. Released via the fusion of multivesicular bodies with the plasma membrane, and the size of the dexosome is 20 to 300 nanometers. And the and biochemical properties distinguish them from the other extracellular vesicles such as microvesicles, apoptotic bodies, and necrotic vesicles. And surrounded by a dexosome are surrounded by a lipid bilayer that contains different type of biomolecules, including proteins, carbohydrates, lipids, nucleic acids, and it is heterogeneous in nature and reflect the phenotypic properties of the cell type from which they are released. And due to inward membrane vagination of the MVB, limiting membrane during biogenesis, exosomes share an outside and topology with their producing cells, which allow physical receptor and ligand interaction with neighboring cells. Although reticulocyte T cells, mast cells, and resting B cells display induced release of exosomes, other cell types such as macrophage and dendritic cell release the exosomes more continuously. in comparison to these other cells. Exosome can induce immune stimulating and immunosuppressive effects based on their membrane bound surface molecules compositions. In recent years, dexosomes have attracted particular interest because they are functionally involved in the activation of innate and adaptive immune response. Dexosome express functionally MSC molecules and co-stimulating molecules such as CD40, CD80 and CD86, and or these are the potent T cell activator during the initiation and amplification of the immune response. Dexosome contain various members of the tumor necrosis factor superfamily such as TNF alpha, FASL, and trial, allowing them kill tumor cell through FASL and FAS trial mediated apoptosis. Dexosome able to induce NK cell mediated gamma interferon secretion via direct binding of TNF alpha to the TNF receptor of NK cells, by which it inhibits the growth of chlamydia within the cell. NK cell activation via dexosome has been demonstrated and can operate through various NK cell activating ligand. Aim and objectives. And the aim of the study is to determine whether and by which mechanism dexosome chlamydia infected DC are functionally involved in the communication and interaction between DC and NK cells during the anti-chlamydial immune response. Methods: the study design and procedure. Type of study: it is experimental study of cell and cell lysate. First of all, cell culture, and then cell culture study by transmission electron microscopy to analyze the internal cells activity, and western blotting is for the study of protein expression, level of protein expression in the cells, dendritic cell and NK cells and epithelial cells. And second is the study of dexosome. Dexosomes, isolation of dexosome. First of all, isolation of dexosome, then time analysis of dexosome, and mass spectrometry analysis of dexosome, then go term enrichment analysis of dexosome, and then RT-PCR analysis for gene expression analysis, and then the flow cytometry and colorimetry to analyze the infected and non-infected differentiation between the expression and different markers to infected and non-infected cells. Immunofluorescent microscopy to check the internal organs, internal change in the cells, and interferon gamma ELISA for the expression, and then final statistical analysis has been done. And the result is that first of all, C. Psittaci infected DC produce large number of dexosomes. To check this, to analyze this, they perform TAM analysis from non-infected and infected DC cells. Here you can see more dexosomes, chlamydia is present in infected DC, and dexosome is released in infected DC cells in comparison to non-infected DC cells. Here they perform production increased amount of dexosome. To identify the increased amount of dexosome by infected DC cell, they perform western blotting, and they identify the marker, flotulin marker, that is present in infected DC, that in comparison present in non-infected dendritic cells. And by chlamydia HSP60 is not present in infected dendritic cells, because that means these cells are infected with chlamydia, and non-infected cell is not infected to chlamydia. To check this, they perform chlamydia HSP60 protein. And here the flotulin is the exosomal markers which is present in higher, the expression of flotulin is higher in infected dendritic cell in comparison to non-infected cells, and beta actin is also high and act as a control. Here is the flotulin signal intensity intensity. In this case, non-infected dendritic cells have low intensity floating intensity in comparison to infected design. Means it means कि infected DC cells produce more dexosome in comparison to non-infected DC. And now they perform dexosome protein analysis by Western blotting and found कि infected DC and non-infected DC more or less similar in protein level, but the expression of protein is higher in infected DC. It means infected dendritic cells produce more dexosome in comparison to non-infected DC. MVB dramatically accumulate in Chlamydia psittaci infected DC cell, and exosomal marker flotulin 1 was present in the supernatant of both infected and non-infected DC, but the level is higher in infected DC cell. Then densitometric quantification of the flotulin signals showed 5 to 6 times higher level in the infected DC samples. More exosomes were released from infected DC than from non-infected control cells. And the quantitative difference is a characteristic pattern of 14 dominant exosomal protein was virtually identical in the two samples, but the expression of all these 14 proteins are higher in the infected dendritic cells in comparison to non-infected dendritic cells. And Chlamydia psittaci infection leads to an augmented release of dexosome, which apparently have a proteins composition similar to those released from the non-infected cells. Now, dexosome released by psittaci infected DC bear apoptosis triggered, but no bacteria. They want to identify कि dexosome released from the infected DC have infected chlamydia or not. To analyze this, they perform dex from chlamydia infected DC cell. There are no TAM analysis for dex from chlamydia infected cell. There are no chlamydia bodies present in the dexosome. And to analyze again this, they perform Western blotting to analyze Western blotting for according to these markers for cell infected DC cells and isolated dexosomes. Now we can find that in non-infected cell and infected cells are both secreted the flotulin, but dexosome flotulin non-infected cells in case of dexosome have less flotulin in comparison to infected DC. Now these FAS and CD19s also present in also less secrete in dexosome in comparison to cells which is infected and non-infected. And GAPDH is also secrete less secrete in dexosome in comparison to infected and non-infected cells. And these are the Chlamydia SHP16 and Chlamydia LPS, which is absent in dexosome. Either it is infected or non-infected. It means कि Chlamydia psittaci is not present in the dexosome, but it is present in infected bacterial cell and not present in non-infected cells. Transmission electron microscopy analysis released vesicular structures have diameters between 30 to 200 nanometers, which match the dimension described by isolated dexosome. And dexosome purified from infected tissues do not contain electronic dense bacterial structure, suggesting that they are free of chlamydia. No Chlamydia psittaci, HSP60R or lipopolysaccharide were present in this material. They also indicate that infected DC do not contain bacterial component. Both transmembrane bound TNF-alpha and Fas ligand were found in dexosome from infected and non-infected disease. Means dexosomes also can also induce the immune response. Exosomal marker flotulin and GAPDH indicate that dexosome may play a role in the induction of apoptosis as well as in the control of anti-chlamydial immune response. Both transmembrane bound TNF-alpha and Fas ligand were found in exosome for infected and non-infected DC, indicating that Dexosome may play a role in the induction of apoptosis as well as in the control of antimicrobial, anti-chlamydial immune response. Now, no chlamydia protein could be detected by MS analysis, confirming that dexosome synthesized and released during chlamydia psittaci infection of dendritic cells do not contain significant amount of chlamydia proteins. Now, we can see कि multiplicity of infectivity 10 level shows more intracellular staining staining in comparison to 50 microliter dex staining and controlled staining. This means MOI 10 have maximum chlamydial body within the cells, and again they identify proportion of infected cells to control, and MOI 10, MOI 5 and 50 microliter dex and 50 microliter 25 microliter dexa. Now we find ki control and 50 microliter dexa and 25 microliter dexosomes have the have less 50 microliter dexosome and 25 microliter dexosome constant amount have similar to the infectivity with the control in comparison to MOI 10 and MOI 5. And MOI 10 and MOI 5 also shows the infection proportion of infected cells depends on the MOI. Here again they perform Western blotting for these parameters, and they found कि chlamydial HSP16 and chlamydial LPS is not present in 50 microliter dexosome and 25 microliter dexosome suspension in comparison to MOI10. MOI10 is present and control is present, and GAPDH is a loading control here. Now, dexosome release from infected DC induce interferon gamma production by NK cells. To identify this, NK cells cultivation with dexosomes and analyzed by Western expression of interferon gamma analyzed by Western blotting. Here we find after 12 hours, 24 and 48 hours, interferon gamma is secreted by the NK cell after DC infection, cultivation with DC infection. And here the release of interferon gamma secretion NK cells also analyzed by 50 microliter buffer controls and the dilution of 50 microliter dexosomes. Here we can identify 1 to 1 ratio is higher amount of interferon gamma in comparison to other dilutions. Here the secretion of release of secretion by NK cell is identified with different parameters, that is NK cells NK cells infected with the dexosomes, the release amount is this, and then amount is secreted, the amount is this, and the dexosome with dexosome the secretion release of secretion level is this. When we then again they incubate with anti-TNF alpha, and the secretion level is significant with the NK cells, and in case of alpha TNFR it also significant with the NK cell, and alpha TNFR2 it is also significant with the NK cells, and isotypes and addition of isotypes of isotypes antibody it is not significant with the combination of NK cell and dexosomes. And dexosome treated NK cells profoundly upregulated the expression of interferon gamma in a time dependent manner, suggesting a strong activation And anti-treatment of NK cells with dexosome indeed cause robust interferon gamma secretion, and strikingly the extent of interferon gamma release correlated with the concentration of added dexosome. NK cell activation is fine-tuned depending on the amount of dexosome to which the cells are exposed. NK cell activation appears to be achieved through the interaction of dexosome bound TNF alpha with NK cell associated TNF alpha receptors. And now the cooperation between DC and NK cell via dexosomes, TNF alpha and interferon gamma reduce chlamydial growth and attenuation of bacterial subversion of apoptosis. Now first of all they prepare the medium, control medium, and dex slash NKUG means the interferon gamma containing culture supernatant was the dex and NK cell are excluded from this. Now they also identify the proportion of infected cells. Here the infected cells with dexosome is level is this above 60, and chlamydia with chlamydia and medium control the level is above 60, but dex NK with interferon supernatant gamma is reduced below 30, and with the interferon antibody antibody of interferon gamma is also non-significant because gamma antibodies reduce the gamma interferon gamma that cannot inhibit the cell Chlamydia psittaci in epithelial cells. And same case is the interferon FINR, because in this case is also non-significant. But in case of isotypes, it become less. And here is the same here, because they won't kick epithelial during the epithelial at the epithelial cell and perform western blotting. The HSP16 chlamydial HSP60 and HSP60 is higher in case of epithelial cells, and chlamydia and medium control experiment in comparison to isotypes. It means कि interferon DC and NK cells cooperate via interferon gamma, and interferon gamma reduce the chlamydial's growth inside the epithelial or other infected cells. Interferon gamma containing supernatant of deoxysome activated NK cells strongly impact Chlamydia psittaci growth in infected epithelial cells. This effect was reversed by the addition of anti-FNR receptors antibodies. Dexosome act as a crucial trigger for NK cells mediated interferon gamma production, which in turn suppress chlamydial growth in infected epithelial cells. Cooperation between DC and NK cell attenuation, bacterial subversion of apoptosis. They want to explain, identify here कि interferon gamma and this DC and NK cell cooperation reduce the apoptosis in the infected cell or not. Now they perform flow cytometry and flow cytometry of infected and non-infected cells, not infected cells. Now the annexin is the apoptotic marker. Here we can see annexin non-infected cells, apoptotic this is the non-infected non-apoptotic cells, and these are the apoptotic cells which have annexin marker. And in case of infected cells, these are the unstaining annexin cells, and these are the apoptotic cells which express annexin V5. And after this, these are the markers which are combination of these markers induce the apoptosis apoptosis in case of infected cells. Here we can see that the interferon gamma and DEX have the apoptotic signals similar to control, but in case of combinations, the TNF alpha and interferon gamma and DEXA and IFN gamma and DEXA, TNF and alpha and IFN gamma, the apoptotic signals will be increase. Here, proportion of apoptotic anoxin V positive cells. We can find the TNF alpha, interferon gamma, and DEXA have significant proportion have similar more or less similar to the annexin positive cells, similar to the control. But in case of TNF alpha and interferon gamma and DEXA and slash interferon gamma, DEXA slash TNF and slash alpha are more significant in comparison to non-infected DC. Here they perform caspase apoptotic markers. For Western blotting for caspase apoptotic marker in all these combinations, and they find then find that TNF alpha, the combinations have more cleaved caspase 3. That means these combinations induce apoptotic cells, as we can see here. The dexosome TNF alpha slash DEXA and interferon gamma, the cleave caspase 3 is less secreted in the infected cells, and TNF alpha, interferon gamma, and DEXA have the similar expression to the control. The treatment of epithelial cells with TNF and dexosomes alone already caused a detectable increase in the number of annexin V positive apoptotic cells, whereas interferon gamma treatment alone had no such effects. However, when simultaneously treated with both cytokines or with a combination of interferon gamma and exosome, a marked increase in the proportion of apoptotic cell was observed. As expected, based on previous study, Chlamydia Psittaci infected epithelial cells display less apoptosis than non-infected epithelial cells. Nevertheless, the effect was observed in both situations, albeit to somewhat different extents. And cleaved caspase 3 specific antibodies confirm this observation that showed that Chlamydia exerts a well detectable anti-apoptotic effect in the infected cells population. And triple combination treatment with dexosome, FN gamma, and TNF alpha caused no further increase in apoptosis. And caspase 3 cleavage also revealed measurable apoptosis after treatment with TNF alpha and dexosome, which in both cases was increased by the additional interferon gamma treatment. And now DC dendritic cell and NK cell cooperate in the anti-chlamydial response. Now they again perform perform experiment by flow cytometry and real cell count after a non-infected and infected cell according to the infection dendritic cell suspension and infected DC and NK cell suspension, and NK cell suspension and DC cell suspension alone. Now we find that in medium the apoptotic signals in combinations is higher than in comparison to treatment of alone NK cell and dendritic cells. And the same situation is here. Proportion of apoptotic annexin V positive cells, medium control, and the combination of DC NK cell suspension and infection DC suspension is higher in comparison to NK suspension and DC suspension. In case of NK suspension and DC suspension alone, it is not significant compared to medium control and infected DC and NK cell suspension. and DC and NK cell cooperate in the anti-chlamydial response. Now they further perform the western blotting of caspase 3 expression, and with the same with the same suspension of markers, that is medium control, infected DC suspension, infected DC and NK cell suspension, NK cell DC suspension. And here we can see कि caspase 3 expression is caspase 3 expression is lower in DC suspension in case of infections, and it is higher in infection in DC NK cell suspension and infected DC not DC suspension. And the proportion of the apoptotic antigen B positive cells in case of these combinations, we can say when the epithelial cells and chlamydia medium control epithelial cells and all these markers, the combination of isotypes combination of isotypes in case of combination of isotypes, it is non-significant and is similar to the epithelial cell combination of epithelial cells, chlamydial cells, medium control cells, and the infected DC NK suspensions. And the treatment of non-infected epithelial cells with culture supernatant from infected DC triggered the incubation of apoptosis. Apoptosis induction was significantly enhanced when instead supernatant derived from infected DC/NK cell culture were used. As expected for infected epithelial cell, basal apoptosis was reduced. Nevertheless, incubation of infected epithelial cell with culture supernatants derived from infected DC for 24 hours caused the induction of apoptotic cell death, which again was markedly increased when supernatant from infected DC and co-culture were used. And infected epithelial cells showed reduced caspase 3 cleavage. Apoptosis was sharply enhanced in the presence of supernatant from infected DC and NK co-cultures. And the flow cytometry analysis of apoptosis induction in infected cells treated with DC, NK cells, supernatant demonstrated that neutralizing anti-IFN gamma and anti-TNF alpha, anti-FAS-L and mixture of these antibodies inhibit annexin V staining with 35, 60, 30 and 85% respectively. And now discussion. And exosomes are membrane vesicles of late endosomal origin and can serve as a means of intracellular communications. And a DC-derived exosome have received much attention because they harbor various immune-stimulating components. And Chlamydia Psittaci infected DC release significantly more dexosome than non-infected DC. And like other types of exosome, dexosome are formed through the inward invagination of the MVB limiting membrane and are released from the cell when the MVB fuse with the plasma membrane. Enhanced exosome released by DC may therefore be a consequence of the infection-induced upregulation of exenophagic degradation. And the infected DC dendritic cell, chlamydia infected epithelial cells do not show infection-induced exenophagy and release fewer exosomes than non-infected controls. This suggests that chlamydia as an energy nutrient parasite affects exosome formation and or release of infected host cells. And redirection of host ATP for chlamydia intracellular survival and metabolic activity depletes energy available for exosome, generates and release. It is possible that augmented release of dexosome is also due to metabolic reprogramming of infected DC. These could allow host cell survival and promote killing as well as exenophagic elimination of chlamydia. In our experiment, no Chlamydia Psittaci protein or intact bacteria were found in purified dexosome or infected dendritic cells. This render it unlikely that they contribute the chlamydia pathogenesis and bacterial spread. And the dexosome from dendritic cell harbor substantial immunomodulatory properties and mediate essential innate immune function, including the activation of NK cells. Fast and trial dexosome also present membrane-linked TNF-alpha on their surface. The interaction of this dexosome-associated TNF-alpha with the corresponding TNFR and NK cells induce the secretion of interferon gamma. Phase and trial dexosome also present membrane-like TNF-alpha and their surface at 63. The interaction of this dexosome-associated TNF-alpha with the corresponding TNFR and dexosome from infected DC also contain membrane-linked TNF-alpha and they induce interferon gamma secretion by NK cell via the TNF and TNFR-alpha signaling pathway, which both play crucial role in the activation of interferon gamma production by NK cell. Our studies demonstrate that dexosome and TNF-alpha in combination with interferon gamma elevate the induction of apoptosis in both non-infected and infected epithelial cells. And the combined effect of TNF-alpha or interferon gamma reduces Chlamydia Psittaci mediated subversion of apoptosis. And synergistic cooperation of TNF-alpha and interferon gamma may have the advantage of rapidly eliminating intracellular chlamydia and infected cells before the bacteria can enter into the persistent state. And these are the postulate working model for the role of dexosome during the chlamydial cell infection. And the discussion about this picture is here. The DC and NK cell are among the first immune cells involved in the counteracting a chlamydia infection and spreading. And both cell type are known to be the less permissive to developmental cycle of chlamydia than other cell types, resulting in impaired bacterial growth and reduced production of infectious elementary bodies in the cells. During the course of dendritic cell infection, increased biogenesis of MVB is directly coupled to the increased formation of release of exosomal vesicles, that is exosomes. And the dexosome contain Fas L and CD95L and transmembrane bound TNF-alpha on their membrane surface and are able to kill epithelial cells by the induction of apoptosis, because these are the apoptotic marker. This is observed for non-infected target cell and to lesser extent for infected target cell. And the interaction of membrane-linked TNF-alpha with corresponding TNFR on neighboring cells, NK cells, lead to massive interferon gamma production and secretion from the NK cells and reduce chlamydial growth in infected cells and synergistically increase the weak apoptotic effect of TNF-alpha and dexosome secreted by infected dendritic cell on both infected cells and surrounding non-infected epithelial cells. And the combined effect of dexosome, TNF-alpha and interferon gamma produced by the early arbitrage of innate immunity may control the cell-to-cell spread of chlamydia at early stage of local infection when the number of pathogens and infected cells is very low. And the joint force of the early defense may thus reduce intracellular chlamydial growth and the number of available living host cells at the immediate site of infection, as well as the subversive chlamydial resistance to apoptosis. Thank you.
Please pose the question if there is any question for Mr. Sonu. So I think there's no question you can close.
No, no, sir. कौन सा बोलो तुमने मिनिमाइज करो एंड को ये यहाँ से बोल रहा था